In 1918, a virus killed more people in 2 years than all the bombs and bullets of World War I. It left scientists haunted by the same question for a century: where did this nightmare originate, and why was it so deadly? The truth was frozen beneath Alaskan ground, its secrets locked inside a single lung. Only now, with ancient DNA, can we finally uncover how the deadliest pandemic in modern history really began, and why that answer changes everything about future pandemics. Brevig Mission sits on the edge of Alaska's Seward Peninsula, a place so remote that in 1918, news from the outside world arrived months late. That November, the influenza pandemic swept in on the wind, reaching the Inupiat village just as winter closed its grip. In a matter of 5 days, 72 adults, almost every parent, elder, and caretaker, were gone. Only a handful of children survived, left to wander through houses filled with silence and bodies, searching for food in the cold. The survivors dug a grave into the frozen earth, working through ice and grief, laying their loved ones together in the permafrost above the village. For these 8 children, the world had shrunk to a landscape of loss. Their memories, later shared with anthropologists and local clergy, spoke of dogs howling in the distance in the sharp, unrelenting cold. The story of Brevig Mission became a warning, carried through generations, a reminder of how quickly a village could vanish when an invisible enemy arrived. Above that hill, the ground stayed frozen, holding not just the dead, but the clues to a mystery that would haunt scientists for decades. Beneath the surface of Brevig Mission, the ground never truly thaws. Permafrost holds its icy grip year after year, acting as a natural vault for everything buried within. In this frozen earth, decay nearly halts. Microbes that break down flesh and bone in warmer soils fall silent, their activity arrested by cold. Lung tissue, especially, fares better than most. When influenza struck in 1918, the virus multiplied furiously in the deep recesses of the lungs, leaving behind a dense reservoir of viral particles. The bodies laid to rest in Brevig's mass grave carried this evidence inside them. For nearly 80 years, the permafrost preserved not just the shape of the lungs, but the fragile strands of RNA that made up the virus's genetic code. Unlike DNA, RNA is notoriously unstable, breaking apart quickly in normal conditions. But in permafrost, the ice crystals lock everything in place. Enzymes that would normally shred genetic material become dormant, and the viral blueprint remains trapped within the cells. Decades later, scientists believed that lungs from these graves could still hold the answers, offering a rare chance to read the genetic instructions of a vanished killer. The grave above Brevig Mission became, by accident, a time capsule for one of history's deadliest mysteries. In the mid-1990s, a new kind of detective work began, not in the frozen ground of Alaska, but in the archives of the Armed Forces Institute of Pathology in Washington, D.C. Dr. Jeffrey Taubenberger, a molecular pathologist, sifted through thousands of paraffin-embedded lung samples collected from U.S. soldiers who died in 1918. These tiny blocks of tissue had been preserved for nearly 80 years, their original purpose long forgotten. At the time, most scientists believed that any genetic material inside would have been destroyed by decades of chemical treatment and storage. But new molecular techniques were changing what was possible. Taubenberger's team developed methods to extract and amplify short fragments of viral RNA, targeting the genetic remnants of the 1918 influenza virus. The process was painstaking. Each sample yielded only slivers of code, and many attempts failed. Still, they managed to recover fragments of the hemagglutinin gene, a crucial piece of the viral machinery. In 1997, the group published their findings in Science magazine, revealing that the 1918 virus was an H1 subtype, with features closely resembling avian influenza. This partial sequence was the first genetic evidence linking the pandemic to a bird-origin virus. The breakthrough did not just answer old questions, it raised new ones. If short, damaged RNA could be recovered from paraffin blocks, then better preserved tissue locked in permafrost might hold even more of the viral blueprint. For Johan Hultin, now reading the news of Taubenberger's discovery, the story of Brevig Mission was no longer a closed chapter. The hunt for the full genome was back on, and this time the tools might finally be up to the task. Johan Hultin arrived in Brevig Mission in August 1997, carrying little more than determination and a promise to disturb as little as possible. He met with the village elders in the school, explaining his hope to understand what had killed their ancestors in the world. Their approval granted after careful discussion, carried the weight of memory and trust. With 4 local boys, Hultin walked up the hill to the mass grave. The ground, frozen even in summer, gave way slowly as they dug through layers of ice and soil. 4 feet down, Hultin uncovered the body of a woman whose size had shielded her organs from decades of freeze and thaw. He would later call her Lucy. Using pruning shears borrowed from his wife, he cut through the ribs and reached the lungs. The tissue was still pink, astonishingly well preserved after 79 years. Hultin placed both lungs in formaldehyde, then sealed them in containers designed to protect fragile RNA from the moment they left the ground. He took care to rebury Lucy and restore the grave, honoring the elders' request for respect and closure. Back in California, Hultin divided the lung samples into 4 separate shipments, sending each by a different courier to minimize the risk of loss. Every package arrived safely at Dr. Jeffrey Taubenberger's laboratory in Maryland. For the first time, scientists had in their hands the physical material needed to read the entire genetic script of the 1918 virus. The bridge from a remote Alaskan grave to a modern laboratory was complete, and the search for the pandemic's true origin could finally move from frozen Earth to molecular code. Inside a high-security laboratory at the CDC, Dr. Terence Tumpy set out to answer a question that had haunted science for nearly a century. Was the genetic sequence alone enough to explain the 1918 virus's lethality? Using reverse genetics, Tumpy painstakingly assembled the virus from scratch, segment by segment, inside a biosafety level 3 facility reinforced with negative pressure, HEPA filters, and multiple security barriers. For weeks, he worked in isolation, donning a full-body suit and respirator, handling what would become the deadliest influenza strain ever tested in a laboratory. When the reconstructed virus was introduced into mice, the results were immediate and devastating. Animals exposed to even tiny doses died within 4 to 6 days. Nearly every single one. The lethal dose was up to 1,000 times lower than for ordinary flu strains. Their lungs teemed with tens of millions of viral particles, far surpassing anything seen with seasonal influenza. It was the virus's genetic makeup, especially its hemagglutinin and polymerase genes, that drove this explosive replication and rapid death. The experiment proved, beyond theory, that the raw instructions encoded in the 1918 genome alone could recreate a pathogen with extraordinary power. Genomic evidence has rewritten the story of the 1918 pandemic's origin and impact. Decades of debate over whether the virus began in Kansas, France, or China have given way to a new picture. The so-called Spanish flu was neither Spanish nor born in a single place. Ancient RNA recovered from victims in Alaska, Germany, and Switzerland reveals a virus that adapted in humans through a series of mutations, likely after jumping from birds, well before the world recognized its danger. By the spring of 1918, the pandemic strain was already circulating on both sides of the Atlantic, carrying key genetic changes that let it spread and kill with unprecedented speed. This detective work did more than solve a historical puzzle. The full genome of the 1918 virus became a reference point for global health. When a new H1N1 strain appeared in 2009, scientists could rapidly compare its genetic features to those of the 1918 virus, guiding vaccine development and risk assessment. Today, the lessons from Breivik mission and the labs that followed shape how we monitor emerging threats, search for dangerous mutations in bird and swine flus, and respond to new pandemics. The tragedy and science of 1918 now serve as both a warning and a blueprint for survival. Now, every new virus faces the scrutiny unlocked by those frozen genes. The 1918 pandemic taught us that the deadliest threats often come from what we cannot see and from what we do not prepare for. In a world of emerging outbreaks, the next lesson may already be waiting in the ICE. Share your thoughts below.